Another class of biomolecules which is very important, that is proteins. As we all know very well that proteins are required for growth and maintenance of the body, and the chief sources of proteins are milk, cheese, pulses, fish, meat, peanuts, etc. Basically, proteins are polymers of alpha amino acids, and we know that amino acids are a class of organic compounds having an amino as well as carboxyl group. You can see that amino group and carboxyl group present in alpha amino acid. Now, amino acids are further classified as essential amino acids and non-essential amino acids. Essential amino acids are those acids which cannot be synthesized in the body, therefore, they must be taken through diet. Example of essential amino acids are valine and leucine. Another class of amino acids is non-essential amino acids. These amino acids can be made in the body, example glycine and alanine. Now let us discuss about Zwitter ion of amino acids. In aqueous solution of amino acid, the carboxyl group can lose a proton and amino acid can gain a proton, giving a dipolar ion having both charges positive and negative, and such dipolar ion is known as Zwitter ion. This Zwitter ion is neutral but contain both negative and positive charges. Therefore, it is amphoteric in nature. So, in Zwitter ion, actually carboxylic group, which is acidic group, वो पॉइंटर नहीं आ रहा था. So, alpha amino acid has acidic group COH and basic group amino NH2 group. So, hydrogen ion moves from carboxylic group to the amine group. So, now Zwitter ion has two kind of ions, NH2 positive and COO negative. Hence, it is known as it is dipolar in nature and amphoteric in nature. Now, structure of proteins. Proteins are polymers of alpha amino acids and they are connected to each other by peptide linkage or peptide bond. So, peptide linkage or bond is an amide formed between carboxylic group and amine group, NH2 group. The product of reaction between two amino acids is known as dipeptide. In tripeptide, there are 3 amino acids, and in polypeptides, there are more than 100 amino acids. So, you can see here the formation of peptide linkage. Carboxylic, there is actually reaction between the carboxylic group of alpha amino acid and hydrogen of alpha amino acid. So, there is formation of CONH, which is actually peptide linkage between two amino acid. So, this is peptide linkage between two amino acids. Classification of proteins. Proteins can be classified on into two types on the basis of its molecular shape: fibrous proteins and globular proteins. Fibrous proteins are product of linear condensation of polypeptide chains and they are insoluble in water. Polypeptide chains are held together by hydrogen and disulfide bonds. Examples of fibrous proteins are keratin, which is found in hair, wool, and silk, etc., myosin, which is present in muscles. Another class of proteins is globular protein. These proteins are product of cross-linked condensation of polypeptide chain. It means there is cross condensation of polypeptide chain. These proteins are insoluble in water. Polypeptide chains folded to give three-dimensional shape and they are attached with hydrogen bond. Example of globular proteins are insulin and albumin. So you can see in picture the fibrous protein, which is actually the linear structure, and globular proteins, which are condensed structure of peptide chain. Now the structure and shape of proteins can be studied at four different levels. We can uh, study the shape and structure of proteins, and these are primary structure, secondary structure, tertiary and quaternary structure. So let us discuss about primary structure of proteins. Polypeptide chain in a protein has amino acids which are linked with each other in specific sequence, and this sequence of amino acid is set to the primary structure. So this specific sequence of amino acids, that is known as primary structure of proteins. So it is the simplest structure of protein. Now the secondary structure of protein. The structure of protein refers to the shape in which a long polypeptide chain can exist, and this structure found in two different forms. The first structure is alpha helical structure. This is the structure of polypeptide chain, and hydrogen bonds are formed by twisting into a right-handed screw with NH group of each amino acid, and residue hydrogen bonded to the CO of an adjacent turn of helix. It means there is actually uh, peptide linkage uh, between the polypeptide chain, and it gives actually the helical structure, helix like structure, so therefore known as alpha helical structure. Another form of secondary structure is beta pleated structure. In this structure, all peptide chains are stretched and they are laid side by side, which are held together by intermolecular hydrogen bond. Actually, this structure resembles with the pleated folds. Therefore, it is known as beta pleated sheet. So you can see here beta pleated sheets in beta pleated structure. So actually folds are found in beta pleated structure. The third structure is tertiary structure of protein. The further folding of secondary structure of proteins give tertiary structure, and this gives two major molecular shapes, fibrous and globular. So tertiary structure can be classified as fibrous and globular. In fibrous there is linear, it is linear form of protein, while globular is condensed form of proteins. The major sources which stabilizes the secondary and tertiary structure of proteins are hydrogen bond, disulfide linkage, van der Waals and electrostatic force of attraction. So in tertiary structure actually it is more condensed form of polypeptide chain. Quaternary structure. Some of the proteins are composed of two or more polypeptide chains, and arrangement of this chain with respect to each other is known as quaternary structure. So actually it is more complicated and more condensed form of the polypeptide chain. Here you can see this condensed form of polypeptide chain. Now in this picture you can see primary structure, secondary structure, and secondary structure is classified as alpha helical structure, beta pleated structure, tertiary structure, and quaternary structures. So these are various structures and shapes of proteins. Now next is denaturation of proteins. When protein is found in biological system with 3D structure and normal biological activity, it is called native protein. But when this natural or native protein is subjected to physical change like change in temperature or chemical change like change in its pH, the hydrogen bonds are disturbed. The hydrogen bonds are broken, globules are unfold, and helix get uncoiled. Therefore, protein loses its biological activity. This is known as denaturation of protein. Example, coagulation of egg white on boiling. When we boil egg, it coagulates, and curdling of milk.